Good afternoon. Today is my great pleasure to introduce distinguished biochemist, a friend, Professor Wang Xiaodong. Xiaodong obtained his bachelor degree from the Beijing Normal University in 1984. Then he went to the US through the Chinese and United States molecular biochemical exchange program known as Hkust. Then he obtained his PhD degree from University of Texas Southwestern Medical Center in 1991. Then he did his postdoctoral training with the Nobel laureates, Joel Gosling and Mark Brown, and also in the UT Southwestern. And then in 1995, he became assistant professor in the Emory University. Just 1 year later, he moved back UT Southwestern as assistant professor in 1996, and quickly become investigator of Hkust Medical Institute in 1997. He was rapidly promoted to full professor, and George McGregor distinguished chair professor in 2001. Only take 5 years from assistant professor to chair professor. Currently, he's a director of the National Institute of Biological Sciences in Beijing, and also chair professor of the Tsinghua. Xiaodong is a world-renowned expert on programmed cell death. For students here, if you learn cell biology, you know his work very well. He has made many seminal contributions to the programmed cell death by chemically identify key components, define the functions, including cytochrome C and apopsone, PAF1, and etc. You will learn more in his lecture. Because of his outstanding accomplishment, he was elected to the member of the National Academy of Science in the US in 2004. And later become foreign member of Chinese Academy of Sciences. In addition, he won many prestigious international awards. Here I named a few. The King Faisal International Prize in the Medicine, Shaw Prize in the Life Science and Medicine, Richard Lounsbury Award from the National Academy of Sciences, and the Heckman Award from the Welsh Foundation, and Eli Lilly Award from the American Chemical Society. Xiaodong is the great academic leader. In 2004, he founded National Institute of Biological Sciences, served as a founding director of the institute, and remained as director until now. With his leadership and the vision, and the institute became the top research institute in China and one of the best in the world. Xiaodong is also successful entrepreneur. He's one of the founders biotech company, Belgium. Now, market worth over $25 billion US dollars. And he also found a few other biotech companies as well. Professor Wang also is the wonderful mentor for his PhD students and postdoc fellow. Many of his trainees have obtained independent positions in China, US, and other countries. Another thing I want to mention here, he's also a marathon runner. So that's one, you want to be successful, maybe test your ability to run long distance. Actually, Professor Wang has been the friend of HKUST and served as advisor, scientific advisor for School of Science for a long time. Tomorrow, I'm not sure I can make an announcement. He will obtain his honorary PhD degree from HKUST. Congratulations, Xiaodong. He will become a HKUST alumnus. Welcome to the USD family. Now, leave the podium to Xiaodong, and he will deliver a lecture, Program Cell Death in Organ-Specific Aging. Welcome to the UST.
Thank you, Professor Xie. And it was way over the generous introduction. Now I have to live up to this. Still, first, thank you for coming to this lecture. And I have been to this, I was in this campus actually many years ago. I always felt like I'm somehow connected. When Paul Zhu was president, I visited him a couple of times in here. And then, I know your president, Nancy, for a long time. And we know each other's science for a long time. I feel like we grew up together. In any case, it's truly a pleasure. I just realized that this is the first academic visit I had since pandemics. Have been stuck in Beijing, and finally I get out. But now I just learned I can no longer go back. So I may be wandering for a while. But in any case, I think life goes on, science goes on. I'm going to share with you, I would like to share with you a scientific story and the scientific direction our lab has been taking in the last 7-8 years. This is not by design. Actually, what get us into this is total serendipity. But as the project progress, it looks like more and more fitting. First of all, I'm getting up in ages, and the study aging become more time urgent, has more time urgency, and also. As I grow up older and older, especially since I pick up long distance running, I'm become much more patient. I know to run a marathon, you just have to pace yourself. This is the key. This is the secret. You just cannot run too far. You get too fast, early on. So the aging research is perfectly fine for scientists like myself at this stage of my career. Study aging, by definition, you cannot run too fast. Every experiment takes time because the animal needs to age. In any case, let's get started. So, as the team says, my lab has been studying. As Ting mentioned, my lab has been studied programmed cell death. We started with apoptosis. It's one, as we learned previously, like apoptosis is the only form of programmed cell death. As the field started to progress, and you will learn more and more about many different forms of programmed cell death. And apoptosis is just one of the forms. Just to get everybody up to the same page and also give you some contrast what other form of programmed cell death looks like, I'm just going to show you a video of what apoptosis looks like. these are cultured cells on a Petri dish. And since we know apoptosis very well, and we are able to engineer these cells so we can specifically induce them to undergo apoptosis. And this is what they look like. They detach from the dish and they undergo this violent dance, so-called, and they break into all these membrane-bound vesicles. And as you see, when the cell dies of apoptosis, their cellular content never leak out. So it's not inflammatory, and it's so-called quiet cell death. That's the reason for every day, we have billions of cells in our body die of apoptosis, we never feel it. If we run into the soccer field and get kicked by the opponent, and you start to feel pain and swelling and almost immediately because your cell died from physical injury. And that is a different form of cell death that is called necrosis. In that form of cell death, the cell membrane will breaking down, cellular content leak out, and you start to get inflammation response. And also for many years, we always thought apoptosis is programmed, necrosis is accidental. But as we learn more, and we also realize necrosis can also be programmed. I'm going to show you in a few slides. So for the field in the last few decades has basically mapped out the pathways, the biochemical pathways of apoptosis. And I'm not want you to remember all the details about this pathway. I just want to remind you that as we know now, the cell undergo apoptosis because they have this intracellular protease called caspase, especially called caspase 3 and 7. These proteases existed in the living cells as a zymogen. They are inactive form. And when apoptosis starts, these proteases become activated and they start to cleave many important intracellular substrates. And these cleavage give this morphological changes as I show you in the video. And for apoptosis, biochemical pathway of apoptosis is basically to figure out how these caspases that exist in the living cells as inactive zymogen, they become activated when the cell decides to undergo apoptosis. And as we know now, clearly, I'm going to show you later, this is clearly not a complete picture, that we have two ways of activate a caspase. One is extrinsic pathway that is triggered by the signal from cytokines. They call the cell death family of cytokines, exempted by the tumor necrosis factor, TNF-alpha. And these cytokines bound to its receptor will result in a protein complex, including caspase 8, at the cytoplasmic side of the receptor. And once these complexes start formed, will also include FADD and RIP1, the caspase 8 will become active. And the active caspase 8 will cleave caspase 3 and 7, leads to the apoptosis. And in addition to extracentric pathway, this caspase activation could also be initiated from the intracellular stimuli, such as DNA damage, excessive DNA damage. And these stimuli will act on a BCL2 family of proteins that consists of two flavors. The BCL2, which is anti-cell death, and they will bind to the protein called BAX. And these BAX, once they are freed from the BCL2, they can punching holes on outer membrane of mitochondria, result in releasing of protein like cytochrome C and SMAC from the mitochondria, intermembrane space. And these proteins, once get out of mitochondria, will drive the activation of caspase 9, also leading to the activation caspase 307, or they release the protein SMAC will bind to intracellular caspase inhibitors called IAPs, and to remove the inhibition of these IAPs on the caspase activity. So as you see, this is a well-designed, almost the circuits that has, the initiator, has the brake, has the anti-brake, and the perfectly good circuits that one cell decides to check out this world, and the circuits become activated, and caspase activated, the cell die by apoptosis. So the story I'm going to tell you initiated from this particular study. This is a collaborative study between my lab and Yigong Xi's lab when he was still at Princeton, that their lab solved the co-crystal structure of SMAC with this IAP domain, a bonding domain. And the structure itself is actually spectacular. And particularly, we realized the SMAC bound to IAP only through these N-terminal 4 amino acid as AVPI. And even the N-terminal 4 amino acid, the true contributors for the bonding affinity is this first alanine, which is only generated when the N-terminal mitochondrial targeting sequence of SMAC get cleaved off and exposed. And also the C-terminal isoleucine, which is provided hydrophobic interaction. The side chains of these amino acid, valine, proline, are actually not required for bonding, so they can be subjected to many kind of modifications. So based on these studies, we collaborated with some chemists and designed small molecules that try to mimic these 4 amino acids. We call the SMAC mimetics. And this is how we designed it. The N-terminal alanine, and you cannot leave it exposed, because if you leave it exposed, it can be quickly degraded. So you need to protect this N-terminal alanine. You can put a methyl group in front. And the C-terminal, because it's provided hydrophobic interaction, you can replace the isoleucine with the hydrophobic moiety of a chemical compound. And you can even replace this peptide bond, so make it more stable at the C-terminal as well. What's really remarkable is we can even link with the linker at the side chain of valine to make it a dimer. Because the SMAC protein itself, native protein, is a dimer. So by connect the valine, you can even link it with proline as well, doesn't matter, to make the dimer, which is function just as the native protein, just as well as native protein. But what's different than native protein is native protein, when it's translated in the cytosol, needs to be transported into the mitochondria. And only when this gets into mitochondria, the N-terminal peptide, signal peptide will be cleaved, then the protein will be active. In the cytosol, with the signaling peptide uncleaved, SMAC protein is not functional because it cannot fit into this bonding groove. But for the small molecule, it will be readily penetrate the cell, and they don't go to mitochondria. They go to the cytosol to immediately take out all the IAPs. So in theory, it will make the cell much more sensitive to any apoptotic stimuli. Truth be told, that this strategy now is being advanced to the registration trials for head and neck cancer coupled with radiation. So, radiation is to induce apoptosis, and the SMAC mimetics is to remove the inhibition of IAPs. So, looks like head and neck combination with radiation will be the first indication the SMAC mimetic will be approved. And the other trial, which is also very interesting, is people use SMAC mimetics to try for chronic hepatitis. Chronic hepatitis B infection, as you all know, is there are many people get infected, and they have risk for develop liver cirrhosis as well as liver cancer. And they use the SMAC memetics, happen to be the cell getting infected and is more sensitive to the SMAC memetics-induced apoptosis. So this trial is also advanced, into registration trials. So SMAC, we often, trying to tell students that sometimes, we working long hours in the lab, we got lost. Why we here? I got a question. How we balance life-work balance, and is there real value that for what we do every day in the benches, pumping solution into different tubes? I just, SMAC memetics is a story. It's a good story, because before we don't even know this protein exists, and we discovered the protein, we discovered how it works, and then we, to try to extract value from what we know how it works, and then we make mimics that now in advanced trials for cancer and the chronic hepatitis B, both major unmet medical needs. So, but we are not talking about how to use it. But for SMAC memetics, when we first designed it, it's become a great 2 molecules. And we start to play with it and by give to all sort of different cell lines and see what it will do. And this will be a typical experiment. Like in this cell lines, which is derived from human pancreatic cancer, we give the cells TNF-alpha, and we are not able to kill the cells because the cells, the circuit is not on because you have IEPs. And we give them SMAC memetics, we take out the IEPs, it's not going to kill the cell because they don't have a apoptotic stimuli. But we put together, within hours, more than 80% of the cells die by apoptosis. Because we know it's apoptosis, we can totally block the cell death by adding a small molecule called ZVAD, which is a pan-caspase inhibitor. So exactly like the diagram predict, like the textbook predict, like it's good, we can write it up, write a paper, very nice. But as science always do to you, is to throw you a curveball. That's when the things start getting interesting. And we did the same experiments on a different cell lines. This is HT29 cell line, which is is derived from human colon cancer. And in this cell lines, we treat same TNF-alpha, SMAC memetics, just like pancreatic cells is the same, it doesn't do much. But what was surprise or not surprise, we put TNF-alpha and SMAC memetics, we only killed this much cells, roughly about 50% of the cells, which is not surprising because some cells are response to particular stimuli better than others. What's really surprising was when we add ZVAD, caspase inhibitor, we not only didn't block what's get killed, the 50% cell killed, we got much more cell killing, as if the caspase inhibitor promote this form of cell death. The caspase is anti-death, so which is totally different than what apoptosis study has been telling us. So this form of cell death clearly is not apoptosis because it doesn't require caspase. It's actually require caspase inhibition. But it's also programmed because they need TNF, they need SMAC memetics. So this is a very different form of cell death. And let's look at it, what does it look like of this form of cell death? Morphologically. This is the same killer cells. They are busted like a popcorn. So they are very different than apoptosis. And they are busted because then their membrane is all broken down, and the cellular content is all going to leak out. So this is the necrosis, but it's programmed necrosis. And also nowadays, people call it necroptosis. It's like apoptosis, but it's a program. So clearly, this is very different than apoptosis. So we spent since 2007, when we first noticed this phenomena, and by the time of 2012, and we basically 2014, we spent like 7 years, and we also deconvoluted this biochemical pathway of this form of cell death, and which is summarized here. And this form of cell death is able to receive stimuli. This is only a partial list, actually, like TNF-alpha, the cell death receptors. And they also receive signal from Toll-like receptors. And many of you know Toll-like receptor is an important part of innate immunity. It's when the bacteria, a virus, in fact, our body, the Toll-like receptor is one of the early warning systems that sense them and start triggering the immune response. And also, there is a molecule called dye that directly sensing the fragments of intracellular, they call the Z form of RNA, and which will be transcribed from particular virus genome. So this pathway is antimicrobial. It's antiviral, it's antibacterial. So it's involved in the immune system. And the activation of these, with downstream of these signals, there are two key components for this pathway. One is this called RIP3, for receptor interacting protein kinase 3. Remember this name. I'm going to say a lot about this. And a substrate called MLKL. MLKL stands for mixed lineage kinase domain-like protein. So just from the name, it looks like a kinase, but it's not a real kinase. So it's a pseudokinase. It has a kinase-like domain protein. And we realize the RIP3 kinase and the MLKL kinase-like can form stable dimers, and the MLKL is a substrate for RIP3. And we map the phosphorylation sites. And we also developed monoclonal antibodies that specifically recognize this phosphorylation site. This is important because now we can use this antibody to specifically go to different tissues to probe when and where this pathway is on. For example, like showed in the bottom part of the slides, this is immunohistochemical staining of liver sample biopsy from the patients who suffered drug-induced acute liver failure. In Chinese, it's called 吃错药了. These people have suffered from acute liver failure. They get rushed to the hospital, and sometimes they took a biopsy. And before, nobody knows how this thing happens. And when we stained this liver biopsy sample with this particular antibody recognized the phospho MLKL signal, you see this massive staining signal. And we also checked the biopsy from some liver transplants, and we never see this thing, the healthy liver transplant. And RIP3, turns out, is also phosphorylated by a particular phosphorylation events, either by another upstream kinase called RIP1, and RIP1 is activated by directly recruited to these receptors. And they can also be activated in a RIP1 independent fashion by a protein called TRIF, and which is receiving signal from Toll-like receptors. So the upstream signal could be diverse, like many signaling events. Upstream could be diverse, so they can receive different type of signals, but converging on the core pathway is RIP3 and MLKL. And MLKL, once they are activated, they can directly go to the membrane and break the membrane and cause necrosis, cause the popping of the cell you just saw. And I mentioned before, this pathway clearly is involved in antimicrobial infections. So the knockout of these two genes, RIP3 and MLKL, as I show you later, does not affect the development as well as fertility of these animals. So they look normal if you don't challenge them with a virus infection or bacterial infection. So this is actually one of the experiments that completely come out of serendipity. It is serendipity because nobody will raise their mouse at up to 14 months, because you need to pay the bills for the housing these animals. And usually you do experiments, you get rid of them, and sometimes you urge your student and postdoc to get rid of them, and you don't keep them beyond, let's say, 5 months. And this story I'm going to share with you actually has a very tragic beginning. The reason we have to keep the animal for long is because the experiments we are planning to do was when the student was already in the third year of her graduate career, and suddenly we saw in one of the prominent journals there's a paper who reported the experiment, the results of experiments we planned to do, and we got most of the results, but we saw their publication, and their conclusion is very consistent with what we saw. So this will be the worst nightmare of any graduate student, that in the third, fourth year of your graduate career, and you just found what your project is just being published in one of the prominent journals. So what do? So in our story, in our case, I can tell you that we end up with a happy ending, but the story certainly takes its turns. That when we saw this, we clearly have a big panic moment in the lab. But in order to calm down the student, I carefully read the paper. I said, they just show the effects of RIP3, has RIP3 or not, but they didn't let the animal live long enough to show whether they have an impact on the longevity. So why don't you leave the animal longer, and you don't have to do a whole lot. You can take a vacation and let the animal see. Let's see whether we can see the impact really has longevity. And a few months later, I forgot about the project. And then she come in and says, these animals look very strange. I said, what happened? She said, these animals, the RIP3 knockout animals, looks very young. And here is what he saw. And this is 18-month-old mouse. 18-year-old mouse, I would say, close to human 70. And these animals, just like human, when they reach to that age, many of them become obese. And it's very cross-the-board phenomena. But these RIP3 knockout animals look lean. And they just look like they are still young. Here is the statistically, they cross the board, looks lean. But there will be many possibilities why animal looks lean, and let's see what else they look different. And then we realize the biggest difference is in this particular organ called seminal vesicles. So seminal vesicle is a part of the male reproductive system. And it's a gland. I'm going to explain to you what the seminal vesicle really is. and it's a gland, so it's a secretive gland. It has a epithelium. It's like a bag, and with liquid inside. And it's known. It's well known when mouse grow older, their seminal vesicles will become larger, and we don't know why they become larger. So we saw this some kind of a compensatory growth, like human prostates. When the old men get old, they have a bigger prostates, and we don't know why they get a bigger prostates, because they probably have this compensatory growth. It's just the thought. But the mouse prostate, they don't get larger, but their seminal vesicles become larger. This is the statistics, but the RIP3 and MLK knockouts, they don't get larger, and they are still looks relatively small, like they were young. Let me explain what a seminal vesicle is. This is a diagram of a male reproductive system of a mouse. And here is the most important part of the male reproductive system, is testes. And the reason is testes is where the sperm is generated. And when they generated, they will transferred and stored in this epididymis. Until ejaculation, the sperm will travel through this tube, then mixed with the liquid secreted from the seminal vesicles. This is the part of the reproductive system become larger when the mouse gets older. And also mixed with this another well-known secretive gland, prostate, and become a semen that get ejected out of the system. So you look at the reproductive system, you clearly realize the most important part has to be the testes, and the rest of them are just, greasy oils. So let's look at what happened to their testes. And before I show you the actual data, I just want to let you get familiar with what testes really looks like. The testes is full of these units called seminiferous tubules. These are the units of testes. A testes will consist many of these units. And these units have this basal lamina as a membrane, which isolate testes from the bloodstream. So testes is just like a brain. It has a barrier. It's just like a blood-brain barrier. It has a testis-blood barrier. So it's not communicated with the blood. So like they say, what happened in Vegas, stay in Vegas, is what happened in testes, stay in testes, which has become very important. So within the testes, they have these two major cell types. One is spermagonia, which are the stem cells, for spermgenesis, and they undergo meiosis to generate immature sperm, which will migrate into the central lumen part of the seminis tubules, which will transferred and stored in epididymis. So in addition to spermagonia, they also have these supporting cells called Sertoli cells. And outside the seminiferous tubules, and you will have hormonal producing cells called Leydig cells, and of course they also have immune cells, myeloid, etc. This is what it looks like when you do a cross-section of the testes, a wild type as well as RIP3 and MLK knockouts. And you see, this is like old testes, like 18 months old testes. And you see in the wild type, you already see the seminiferous tubules, which looks empty. And you see these basal membranes of seminiferous tubules. But in RIP3 knockout and MLK knockout, even the mouse at the same age, you see the thickness of these cell layers of seminiferous tubules, And you also see mature sperms in the lumen of these seminous tubules. And you don't see many like this wide open seminous tubules like in wild type animals. So we also remember we have monoclonal antibodies that specifically recognize the phospho-MLKL, indicating the activation of the pathway. So we also stained, using this antibody to stain the 4 months and 18 months old mouse testes. As you see here, as in wild type animal, at 4 months, you don't see any signal. But when the mouse reached 18 months, you start to see these prominent signals at these cell layers that in the surrounding region of the seminous tubules, indicating there are this form of cell death happening in these cells, including, I don't have time to show the actual data, in both Sertoli cells and the spermogonias, there's activation of this programmed cell death, and which may contribute to the opening up of these vacuoles. But in RIP3 knockout, you don't see it, and clearly, you see the normal-looking seminiferous tubules. And we also got some, work with the people in study doing the surgeons doing the prostate cancer surgery, and we also get some samples from testis biopsies from old men who suffer from prostate cancer. In prostate cancer, the treatment sometimes is to take out the testes because prostate cancer need androgen to grow. And we also get the testis samples from young people who suffer from testis, they call the testis torso, meaning one side of the testis get physically injured. And you have to take them out. If you don't take that part of it out, they become necrotic. Necrotic, it also affects the health of the other side of the testes. So then you can get sample from young and old testes of men. And clearly you see that in a young man, 30 years old, and you see this just like a young mouse. This is seminous tubules, and you see the thick cell layers. But the old man, like 80 years old, and in many of these seminal tubules, it looks dilated, and the cell becomes sparse, and you have this big vacuoles in the middle. And if you stain with phospho antibody, MLKL antibody, you pick up these positive signals in the old man, but not the young man. So looks to us, this pathway is evolutionarily conserved between man and mouse. Remember, I didn't say mammals, because in some mammals, this pathway is not conserved, which is an interesting topic for another day. And so far, I showed you the animal without RIP3 or MLKL, when they grow old, males, and they look young. They are physically look young, their testes look young. Also, I didn't show you the testosterone level. Their testosterone level also remain high. But then the question you often ask, can they still do it? So, meaning that can they still produce progeny? And in mouse, we actually can do the experiment to test it. So we give these 16 months old male mouse, wild type RIP3 knockout, MLKL knockout, 2 female 10 weeks old young female mouse, and we see whether they can still get them pregnant. And then we do the data collection and statistics. So for 16 months old wild type animals, only 3 out of 14 can still reproduce. But for RIP3 knockouts and MLKL knockouts, almost 80% can still produce versus wild type, which is already down to 20%. When they are young, almost 100% can reproduce. So clearly, they not only look young, they can also function as a young animal. But what's the catch? You have to ask, what's the catch? The catch is the babies they produce, which they don't supposed to. Almost all of them has birth defects. So, which reminded us why two things. First is clearly the testis aging is programmed. And its needs and its service purpose, the purpose looks like is to get rid of the old DNA actively from the gene pool. To prevent, as you all know, the old DNA will accumulate mutations. And this program triggers the elimination of old DNA to prevent them to get into the gene pool. If they do, they will produce these defective offsprings. So we believe that is an evolutionary advantage to have this programmed organ-specific programmed aging. Then the question becomes, is this sex-dependent? What about the females? So I'm going to switch gear a little bit and tell you the story about females. Just to give you a warning, and as you may already know, first of all, female is very different than male, is totally different than male, and the female is much more sophisticated. So let's look at the ovary from the female mouse. This is a young ovary of wild type, RIP3 knockouts, and MLK knockouts. And you don't see much difference. And these vacuoles are follicles. When the egg start to mature, they grow into this mature egg, different stage of mature egg follicles. And then, as many of you know, when the time to ovulating, these egg, the cells surrounding the egg, which called granulosa, the granular cells, will die and spill out the egg, and which will, pass through and get if the animal get chance to meet the male, they get fertilized, and you see all these eggs, but at the 14 months, you see the wild type, as well as the MLKL knockouts. You don't see much egg follicle. First of all, the organ becomes smaller, and they look much more fibrotic, filled with the collagens and fiber. But for the RIP3 knockouts, they look a little bit different, and you still see quite a few healthy-looking follicles. And not just the follicles. This is the quantification. This is not like the male male testes that 14 months old, 18 months old, they all look almost totally young, and they still have a major drop. But relatively speaking, the wild type and MLK knockouts has a much bigger drop. These are the estrogen level, E2. And clearly you see from 4 months to 14 months, you will see statistically drop, both wild type and as well as MLK knockouts. But you look at RIP3 knockouts, their drop is not obvious. So they seem to maintain their estrogen level. Then how about the reproductivity? And again, this time we give every female a stud male, young stud male, 10 weeks old, and we see whether the female can get pregnant. You see, at 4 months of age, all these animals, 20 out of 20, can all get pregnant. At 14 months, the wild type, only 23% can still get pregnant. But RIP3 knockout at the same age, almost still 70% can still get pregnant. MLK knockouts, again, down to 25%. So obviously, the RIP3 knockout animal has more egg follicle, and they can still have, many of the high percentage, 70% of them can still have pups, even when they are 14 months old. So again, you probably can guess, all of these pups are not healthy. So we believe in female, the programmed aging also exists, also serve the same function. But the question becomes, all ovary is more complicated. What is truly being triggered here? What is the signal, that aging signal is being sensed here? So let me tell you the rest of the story. We also tested the reproductive longevity. If you don't know what that means, is we keep giving different age female young stud, and we measure at what time they no longer able to reproduce. So clearly, the RIP3 knockouts is significantly longer than wild type. So to figure out what RIP3, first of all, is unique. MLKL doesn't play any role, so this is what we saw. And how RIP3 play a role here? And is that clearly not through this programmed necrosis pathway? And what pathway it goes through? So first of all, we just checked the RIP3 expression in the ovary. We did both Western blot as well as immunohistochemistry staining. And of different aged ovary. And first of all, clearly, you see the RIP3 expression is bell-shaped. It is high in the beginning. This is 2 weeks old mouse. And it gets lower when the mouse becomes sexually mature. And then it's become higher again when they start getting old. So it's bell-shaped. But when we look at where they express, and they obviously look different. One, when the mouse was young, 2 weeks, 4 weeks, 6 weeks old, you see it's expressed in the granulus, the surrounding egg. That's why you see all these rings. When the egg follicle develop, they first from primordial egg, which is like this small clumps. Then when they start to develop, these are granular cells that surrounding the egg. And then the egg starts to develop into fully mature follicles when they intersexually mature. And you see they very nicely stain outside the ring. And here is more clear. But when the animal gets old, let's say 4 months, you start seeing more and more these clumps. And in the end of the day, at 14 months, they all become these clumps. And these clumps are no longer co-localized with the egg follicle. You see, before they always with the rings, with the follicle. Then at 8 months, you see all these clumps. As this here is the egg follicle. It's no longer with the follicle. So, what this looks like there are 2 waves of RIP3 expression. the first wave is the one with all the granular cells that surrounding the egg. And the second wave, it seems to come out of nowhere. And it clumps into this clumps in the ovary. And then what we did is for these, old ovary, the clumps, and we suspect this may have cell death happening. And we stained with many cell death markers, but we found active caspase-3 is perfectly co-localized, with the RIP3 signal. So here is the quantification, and looks like the RIP3 signal that later emerged induced apoptosis in ovary. And this is not just, again, only happening in mouse. So we're able to collaborate with the doctors, in Beijing, study this they called, I can never remember the name. There is an ovary disease. They develop many fibrosis, in the ovary, and then they lost their estrogen level drops. The women start growing hairs on their face and etc. And we got a biopsy from these ovaries, and again we stained active caspase-3 as well as RIP3. And we found, again, these clumps of caspase-3 is co-localized with this RIP3 signal. Here is the quantification. And the location, the location of these signals are in corpus albicans. And the corpus albicans is just, I think is defined by its looks, right? They just look like, it's dead ovary follicle remnant. So looks like the RIP3 has two waves, and the second wave is coincident, co-localized, co-resides with the active caspase-3. So then the question becomes, what really happened? So to dissect biochemically, we cannot study ovary as an organ. We have to bring that, to break down into cell line. So we first just get a cell lines, they call this KGN cells, which is human cell lines derived from the egg granular cells that surround this ovary follicle. And in these cells, this cell actually doesn't express RIP3. But if we express RIP3 artificially with the Dock-inducible expression, and we showed whenever we start expression RIP3, and we see cell death. And the cell death is apoptosis because if we add ZVAD, which is caspase inhibitor, we can prevent the cell death. And we also add this programmed necrosis inhibitor. We discovered, we named NSA. This doesn't able to affect. So this is all apoptosis. When you add, this is a RIP1 inhibitor doesn't affect. So whenever you inhibit the caspase, it's blocked, this form of cell death. So this form of cell death is apoptosis, but it's RIP3 dependent apoptosis. Remember, this is induction of RIP3. So how RIP3 is able to induce apoptosis, so which is become very interesting. Then we start to look for different clues. And so we put, again, RIP3, TAC-inducible RIP3, in MCF7 cells. MCF7 cells is from breast cancer. Again, it's a hormone, estrogen-dependent cell. And we also put in RIP3 into HeLa cells. A HeLa cell usually don't express RIP3. But if we put in, HeLa cell doesn't undergo this necrotic cell death either, programmed necrotic cell. But if we put RIP3 in, they can do it. So we put this RIP3, but remember, necrotic cell death need addition of SMAC-mimetics and TNF-alpha. By simply induction of RIP3 expression does not kill HeLa cells, but it kills robustly MCF7 cells. Then the question becomes, same RIP3, they express in two different cell lines. They're all from female. One is cervical, one is breast. What different they look? Here is different. It looks like in MCF7 cell, you get multiple bands of RIP3. And in HeLa cells, you only get one band. So which telling us they may be phosphorylated in MCF7 cells, and the phosphorylation is important for its function, for induced cell death. So we then go ahead and map the phosphorylation site. And before I show you where the phosphorylation site is, and whether the phosphorylation site is truly relevant, I also want to show you that we also did IP experiments, from MCF7 cells, since we know expression RIP3 will induce apoptosis. And when we pull down the flag, which is tagged RIP3, and we not only, we pull down caspase-8, we pull down FAT, and we pull down RIP1. So it looks like the RIP3 in these MCF7 cells formed a caspase-8 activation complex that is also have FADD and RIP1. If we add caspase inhibitor, we did prevented the activation of caspase-8, which seems to be happened in this complex. Is that true? If they do form this complex internally from RIP3, they will require these components. What about we knock out these components? So we knock out RIP1, we knock out the caspase-8, we knock out FAT, and we also knock out C-flip. C-flip is the caspase-8 inhibitor, which is, they look like caspase-8. They can also recruit to the caspase-8 side, but they don't have activated enzyme. So this is an inhibitor. So you knock out both RIP1, caspase-8, and FAT, the cell is totally saved. These cells are no longer able to die, even you induce RIP3 expression. But you take out flip, it doesn't matter. In a matter of fact, the cell dies faster, so which is consistent. That tells us that when RIP3 was expressed in MCF7 cells, but remember, not in HeLa cells, they will form this caspase-8 activating complex. And they require all these components to activate cell death. So then the question becomes, if we take out FADD, do we also have an ovary phenotype? But the question is, you cannot take out the FADD. If you take out the FADD, the caspase-8 will not be able to activate throughout the development. The caspase, remember, the caspase especially caspase-8, is the inhibitor of this necrotic cell death. And you need to add a caspase inhibitor to induce this necrotic cell death. So same thing that if you don't have caspase-8, the mouse die at embryonic stage, perinatally. Not perinatally, prenatally. The reason is this necrotic cell death happened, in the vascular system, so the animal wasn't able to develop. So in order to get mature animal to study their ovary, you need to same time knock out MLKL or RIP3. And then you get fully survived animal. And we look at animal at 12 months old, and 11 months old. The reason we need to keep them, to study them at a relatively young age, 11 months instead of 14 months, because if you knock out this caspase-8 fat pathway, when the animal get old, they develop this T-cell lymphoma. So we cannot keep them too long. But at 11 months old, it's old enough. And for 12 of them, wild type and FADD MLKL double knockouts, and we give them start male mouse. Wild type only 4 out of 12 can still produce. But these fat MLKL double knockout, 10 out of 12 can still reproduce. Remember, MLKL knockout in animal doesn't affect their fertility whatsoever. Again, the pup they are able to reproduce is not healthy. Same thing as RIP3 knockout. So here is come the model. We believe there exists of a pathway, of apoptotic pathway, intracellular apoptotic pathway, that initiated from RIP3. And they require RIP1, FADD, and caspase-8. And this pathway actually been hinted before that, when Vishwa Dixit from Genentech did one experiment, he made a knock-in animal for RIP3 kinase dead. And then he found the animal wouldn't be able to survive. And this mutant RIP3 actually bound FADD, RIP1, and caspase-8. So he said this apparently activated this pathway. But they never know this pathway actually naturally happening, and it's naturally happening in the ovary. I'm going to show you more. And it's naturally happening because we believe this RIP3 must have a special modification. And I showed you already that happen in FCF7 cells and in granular cells, but not in HeLa cells. And we just need to map out the difference modification in these different two cells, and we know what happened in RIP3 to trigger this pathway. To cut a long story short, and we found in RIP3, serine 164 and threonine 165 are phosphorylated. And we also developed the monoclonal antibody against this phosphorylated RIP3, as you see here. The wild type, if you express RIP3 in MCF7 cells, you pick up the signal by the anti-phosphoryl RIP3 antibody. And if you do mutant, you don't pick up the signal. And with flag antibody, you don't see the upshift of the RIP3 band. Here is in the granular cells. It's the same thing. Very clear. It's just like MCF7 cells. But in HeLa cells, it's very different. Here is the flag. So you express RIP3 in MCF7 cells, the KGN cells, you see 2 bands. But in HeLa cells, you only see 1 band. So HeLa, and you use anti-phospho antibody, you only pick up signals from the cells that is able to undergo apoptosis, you are not able to pick up the signals from the cell unable to undergo apoptosis. So we realize this specific phosphorylation is a signal for apoptosis. And we also know this phosphorylation is due to autophosphorylation from RIP3. So here is the signal. But if it's autophosphorylation, what induced RIP3 expression and what caused them to undergo this autophosphorylation? So here is the answer. Before I show you the answer, now we have this anti-phospho-RIP3 antibody. We go to the ovary of different age and probe it again. Remember, if we just do the RIP3, normal RIP3 antibody, we see this bell-shaped curve expressed high early, but it's all in the egg follicle, the surrounding egg follicle. And then the bell-shaped come back again in later life, and it's all clumps into this unstructured place. But for phospho-RIP3 antibody, only pick up signal when the animal is old. So clearly, there are two forms of RIP3, and one in the late life, RIP3, when expressed, they are phosphorylated. And they don't pick up the follicle RIP3, they only pick up the slumps of RIP3. So here is we even use this antibody versus a transgenic animal, which is tagged with flag. So we are able to stain these two forms of RIP3 simultaneously. And we clearly see, these general RIP3 will still surround the follicles, but the clumps of these RIP3 at the corpuscle beacons, called the corpus region, only the phospho-RIP3 is expressed. Then the question becomes, what signal that induced the second wave of RIP3? And then we start to look into the literature, and we realize it's been reported before. Phosphoglandin 2-alpha expressed in ovary, and look at as the ovary age, its level increased dramatically. And we stained the RIP3 flag with prostaglandin 2-alpha receptor. And we found in the late stage, they co-localize at corpuscle beacon. So look like this tells us prostaglandin 2-alpha might be the signal for late stage RIP3. Is that true? To prove that, what we did was to use prostaglandin 2-alpha analog called we call the DP, DT. And in this experiments, we first induce ovulation, super ovulation in animals to let them produce a lot of egg, to mature a lot of egg follicles. Then we inject DT, and we look at what happens. Here is the staining of RIP3. And with DT injection, you see lots of RIP3 get induced. And what's really interesting is, if you induce RIP3, this is now in young animals, you induce RIP3, the RIP3 can be picked up by phospho-RIP3 antibody. The level also increases. And this is due to prostaglandin pathway. The prostaglandin pathway induces RIP3 through, they activate the ERK-MAPK pathway. And because of time, I didn't have time to show you. If we knock out prostaglandin 2-alpha, the animal looks fine. but they no longer have this phenotype. And again, we showed that it's activate caspase-8, this analog, using the immunohistochemistry, and we show this DT induce caspase 3 activation, active caspase 3 in wild type but not in RIP3, and FADD MLKL double knockouts. Here is the quantification. So the prostaglandin is the trigger, and they through RIP3 the FADD pathway. Here is my final summary slides. I know I'm running slightly over time, and because I want to give you a full story. So we believe for the animal, in animals, not all organs are aged equally. The reproductive organ aged first. This is the elephant in the room. We already know that, right? They age first because they aged through a program. But the male and the female are different. And I'm not claiming it's a full story, but it's it started emerging as a story. For males, it's through TNF family of cytokines. And we believe it's due to accumulative oxidative DNA damage from young to old. Once this to a certain stage, they will activate NF-kappa B pathway. Remember, what happened in the testes stay in the testes. And they will generate paracrine signal and triggered the activation of a TNF family. And in the spermogonium and the Sertoli cells, there are very little caspase-8, almost non-detectable. So naturally, if you activate this pathway, you trigger necroptosis. And with RIP3, MLKL RIP3, and these are the specific phosphorylation events, and they cause the necroptosis of spermogonium stem cells. And this is what causes aging of testes. And then there is secondary consequences like the enlargement of seminal vesicles and the death of hormonal-produced Leydig cells. But for females, as the animal grows young to old, they accumulate these prostaglandin 2 alpha in the ovary, which is the inflammatory signals. The inflammation causes aging, and this is very true here. In this case, the prostaglandin, when it accumulates, they activate the receptor through MAPK pathway, they induce the RIP3 expression. And this is happening later in life in animals, and they happen in the granular cells. And the induction of RIP3 will autophosphorylate itself, and then they form this apoptosis caspase-8 activating complex, and causing apoptosis of regular lutein cells of ovary. And that accelerated, these are the nurturing cells, hormonal producing cells for the ovary. And these cell deaths cause aging of the ovary. And in human, we call it menopause. Lastly, I want to acknowledge the people who actually did the work. This is a graduate student whose project gets scooped. And like I mentioned, the whole story has a tragic beginning, but end up with a happy ending that switched all my major research direction of my lab from cell death now to organ-specific aging. I believe this will take us quite a few years and extended my not reproductive longevity, but my scientific longevity, hopefully for a few more years. And of course, she graduated from the lab, got a postdoc position at the HMI lab at Stanford. Li Dianrong is another graduate student, clearly who also graduated, who take over her project after she left, and also started the ovary project, and figure out the ovary granular cell apoptosis is part of an ovary aging program, only partly, and this has nothing to do with. We believe there are many programs for aging of the ovary, is particularly in egg, but we just don't know what happened there. So I'll stop here and thank you all for listening, and thank you all for your patience to stay here late. Thank you.
And somewhat it's more like in a philosophical level. So I think everything has a price or consequences. If you want to live young and keep fertilized abilities in an old age, the offspring might be unhealthy as the consequence. So my question is, so why this RIP3 is tackled at the seminal vesicle or the ovaries in the mice, and does other organs share the similar pathways or express the similar molecules?
Very good question. We tested all the major organs. We didn't see any difference. And the RIP3 normally is expressed in the epithelial layers that line our GI tract. That's why you see the HD29, which is a colon cancer cell, they express RIP3. And also all the immune cells that express RIP3, T cells, B cells, monocytes, they all express RIP3. So in normal situation, RIP3, we believe, is part of the immune system that give up the organ damage signal. So because many bacteria and viruses, they carry their own caspase inhibitor. So when they inhibit the host, they want the host to live longer so they can reproduce. And otherwise, the host will trigger apoptosis, then they lost their chance. So but when they inhibit the caspase, the GI tract, the immune cells have RIP3. Then if you block caspase, RIP3 come in, they trigger necrosis, they popping the cell open. And then if you cellular content leak out, then your immune system know where there is the infection, there is a problem. And they mobilize all these macrophages, monocytes, the immune system to come here and to attack. So this is how they function normally. But in major tissues, and let's say you take them out, they look fine until you challenge them with like a dose of virus, and then they cannot handle. But normally they look fine. And we look through the brain, lung, liver, the intestines, muscle, and they are no different. Of course, like muscle and liver, and the liver is still express RIP3, like brain express very little RIP3. The lung epithelial also express high level RIP3, and we believe this pathway will play a major role in antiviral that come from respiratory tract. And in matter of fact, we did the RIP1 inhibitor on in a severe case of COVID patients, and we see very good results of alleviating the respiratory stress and save the hospital stay.
Thanks, Professor Wang's talk. Very wonderful. I'm wondering whether there are any defects of RIP3 knockout mice, because if I'm correct, the RIP3 knockout mice can keep young and can reproduce in elderly time, although the newborn perhaps can have some problems. But overall, it seems that the benefits are more than the drawbacks.
Depending on which perspective you are looking at. When they are young, the RIP3 knockout produce normal pups. And when they are old, the wild type is no longer able to produce, and they can still reproduce. And then their pups all have birth defects. It's not normal. But normally we don't keep them that long. And only when your project gets scooped, you have to keep it long to finish up your project, then you keep them that long. Before, nobody keeps them that long. And that's why it's accidental. And it's I would say it's irrelevant in the wild. In the wild, you want to eliminate them quickly, and this thing will act active. When animal get to a certain age, they can no longer reproduce. What's really interesting, I mentioned now you're asking a little bit, but I didn't go into, is this pathway is not conserved in canine species. Like the wolves, the dogs, the tigers, the lions, they don't have this pathway. So, we are joking, these animals, they don't die, they die young, they don't die old. They fight to death. It's like old Chinese saying, heroes and beauties die young because they don't want people to see them old.
Okay, and another question is a very general question. From the study of programmed cell death in organ-specific aging, from your understanding, is there any hints that we can use to slow down the aging process?
Well, this is another philosophical question. I think scientifically, in human we don't know, but for mouse, definitely. I didn't show you we now able to feed these animals, one of the RIP1 inhibitors we have, for 2 years. The animal on normal diet all look very old. 2 years, 2.5, more than 2 years old mouse is like 80 years old human. They look old. They are, their posture is. You look at them, they are old. But we can keep the same littermates on our compounds. Looks very young. No difference from 2 months old. And not only they look young, we even put them into exercises, roller running, water mates. They all perform just as well. So scientifically, we can. But what price is paid, we don't know. We are still studying. And because the animal, we don't know. They look young. They could be they are not stupid. They are also smart. They are young, they are lean, they good looking, they are smart, they move fast. But they could be other unseen. They probably not able to survive COVID. So there's always a balance. There's always a give and take. But we need to understand it. This has become real science. I always, I believe, we are, aging is just like other physiology. It's just like development. It's just reverse of development. And if we now can understand development at the cellular biochemical level, why cannot we understand aging at the chemical biological level? But if it's just wear and tear, it will be difficult. And now I'm telling you, it's not wear and tear. It's program. If it's program, we can understand. We're not only able to understand it, we may be able to even to interfere.
Okay. For time's sake, I have one question from the anonymous student on the line. The question is, in the young mouse ovary, does RIP3 also regulate programmed cell death?
Oh, that's a very good question. This is one puzzle we don't understand. In young ovary, it's expressed, and it's also in a granular cell surrounding the egg follicle. But we don't know what's its function. And the RIP3 knockout, clearly they are knocked out, they have no longer there. The egg function is fine. They still produce, pups. So that is still a mystery for us. We don't understand.
So RIP3 is involved in both necrosis and apoptosis. So do you think
Correct.
there's a reason why evolution choose to use the same molecule in these two very dramatically different death pathways?
Yes. This is, again, good and philosophical question. We know for a fact they can do that, which I didn't go into the molecular details. I even know why, why they choose these two. Like in Hkust cells, you can express very high level RIP3. They don't go apoptosis, but they go necrosis. In MCF7 cells, you express same level of RIP3. All the cells get killed by apoptosis. And we believe the reason is the HSP90 chaperone level. If the chaperone level is high, the sudden expression of RIP3 will be folded correctly. And after they folded correctly, they are inactive. They have to be active by upstream, adapter proteins and the kinases. But if the sudden expression and also the chaperone is low, the RIP3 will phosphorylate itself. They cannot fold it quickly and correctly, and they phosphorylate itself, we believe, as a signal for the cell is no longer able to handle all this. And then if you phosphorylate that particular site, 164 and 65, then it's able to recruit RIP1 and FADD caspase. So there is a switching because of the cellular contents, and the cellular contents is chaperone level.
That's very interesting. So it could respond to the protein misfolding state.
Yes.
Too.
Yes, protein unfolding state, correct.
Thank you.
Xiaodong, very fascinating talk. So for, a related question, what is the mechanism underlying prostaglandin-induced RIP3 increase? It's a transcriptional increase?
Transcription, yes, that is clear.
I see. So you are saying for RIP3, let's say in young animal versus old, even though the protein are still there in the female case, but the obviously in the young in the granulocytes, from the younger mice, they are not phosphorylated, right? The proteins are there, but
correct.
So you think it has something to do with the chaperone level?
Yes, it's also chaperone.
I see. Thank you.
And because we know it's HSP90 chaperone, now there are lots of HSP90 inhibitors.
For time's sake, the last question.
And shift with the inhibitor.
Thank you so much. So I have a question about, I think you touched upon the fibrosis and fibrotic tissues, and I know like PGE2 also has a role there as well. So I'm just curious, do you think this MLKL independent RIPK3 pathway has some connection in the fibrotic tissues or in fibrosis?
That is a good question. And so far, RIP3 clearly can induce apoptosis independently. Whether they can induce necrosis independent of MLKL, at least in the system we look at it, is no. But I also mentioned, like dogs, wolves, tigers, they have no MLKL, but they still have RIP3. So what is their substrates? Do they have MLKL equivalent? or do they just don't have any, this male-mediated, male reprogram aging because they die young by fighting, and they don't have this MLKL, then they fighting, they get easily infected and then they die young. Their wound healing does not look good. So there's also alternative possibility that in these animals there is a MLKL equivalent, but they just don't look like MLKL. The MLKL clearly is a late evolved molecule. So it has a kinase-like, which is serve as a bonding to its upstream kinase. And it's also N-terminus, it's all helix, it's like all helix bundles. So these are the helix bundles that can disrupt membrane. So they don't have sequence homology. So whether they truly have homologue in these different animals. So we really want to get our hands on a dog cell line, and we couldn't find any. Does anybody know there's dog cell line? Let me know. Or wolf or tiger.
Okay, let's thank Professor Wang for the wonderful and inspirational talk.
Thank you.